So what I want to show here is a 10-year record of the dynamics of chlorophyll, which is a proxy for biomass of phytoplankton in the ocean. Now what you notice is there's a highly dynamic system. It ebbs and flows. Also, the productivity in the ocean is turns over in time frames of few days, unlike the trees on land. So it's a very sensitive system because the carbon that's fixed by the algae in cyanobacteria and algae in the ocean, it is rapidly turned over. So the question is, what is the fate of the carbon that's fixed into the ocean? The question is also important because half of the primary productivity on the planet actually occurs in the oceans, and half of the anthropogenic CO2 that we are putting into the atmosphere also ends up into the ocean. So its fate in the ocean and its exchange with the atmosphere is a very important question for the future of the planet's climate, as well as the health of the ocean and its relationship with human affairs. So what I'd like to do is to impress on you that this is a very complex problem, and the question that has been asked and is being asked, and I will ask here, is, so what are the biological forces that create these patterns of productivity, patterns of carbon flow in the ocean and its exchange with the atmosphere? Historically, it was thought that it is animals that eat the phytoplankton, they're eaten by the fish, but now we know that microbes do a lot of it. In fact, microbes are responsible for a large proportion of the flux patterns that emerge from as a processing of this productivity. And however, we still don't know how they do it. We actually have only just an inkling of how they do it. We know how fast they do it, but we don't really know how microbes interact with the productivity in the ocean. So the approaches that we can use have given us a general broad brush picture. Okay? But what we're hoping is that perhaps other approaches can help. And so what I'll discuss is that systems biology may be one approach, which combined with the traditional approaches that we have been using could tell us more about our understanding of the interactions of the microbes with the ocean systems. A drop of seawater off Scripps Pier during a bloom of a red tide dinoflagellate. What I wanted to show here was that the interactions of microbes with at the micro scale is very dynamic and responsive to the productivity events and to the production of organic matter. And that was during the bloom, but this is on a boring, run-of-the-mill day off Scripps Pier. But in any event, when you go swimming, remember that there is a bacteria. When I say microbes here, I'm talking about heterotrophic bacteria and archaea that interact with the organic matter. So be aware that every 100 micrometer on average, there is a bacterium or archaea cell. Okay? So it's a very busy scene. It's a very intimate scene in the ocean. But not so busy. Oh, yes. Okay, right. So what I'm going to talk to you about is exactly this question of interactions of bacteria and archaea with the productivity in the ocean. And what's really we have studied so far is we have good inventories of the microbes' genetic diversity, but we don't really know the function, and more than anything, we don't know the interactions. Okay? We don't know what interactions are occurring, and we don't know what conditions that, for instance, climate change may affect those interactions. Okay? Now, what I've called here microbe pervasively and intimately interact. I'll just read this to you because this is what I'm going to tell you. Okay? But microbes pervasively and intimately interact with all facets of marine ecosystems at very small scale to dynamically structure the global ocean carbon cycle. Complex ecological interaction at these minuscule ocean scales and their biogeochemical consequences remain unexplored. Okay? And this is a big challenge, and we need approaches to understand what are the processes, what are the interactions, and what happens at the very small scale in the ocean. Ecosystem perturbations, for example, climate change, may alter the microbes' interactions and ecosystem structure at this small scale, and it may happen unnoticed by us. So it is possible that the very small scale of the ocean is changing, has changed to a degree that we don't know, because we don't have the techniques, we don't have the tools, we don't even have the intuition at that scale. Okay? So we may be changing the ocean while we think that we are doing all these surveys of the ocean and understanding, making catalogs of what is there, but we may be changing the architecture, the microhabitats in the ocean, which may change how microbes structure, how microbes regulate carbon fluxes, how microbes sculpt the ocean's ecosystems. And you saw those patterns of productivity that recur. Okay? Whether we will change the system in a way that may be catastrophic, we don't know that. Okay. So that's really the main challenge that I'll talk to you about. And my thesis here is that bacteria are major contributors to the ocean ecosystem, what I've called molecular connectivity. And the statement of the obvious is that microbes only interact with molecules at the molecular level. Okay? So I've always had this problem that I could think about microbes activities in the system, but I had problem in throwing in the fish and animals and plants into this ecosystem. Okay? Because, you know, how do you go to deal with this microbes and fish? Okay? And when you think about the molecular interactions, which you reduce the problem, okay, to molecular interactions, okay, then whether a microbe interacts with a swimmer, with a whale, okay, with a phytoplankton cell, okay, or a piece of detritus that is sinking through the ocean, these are molecular interactions. Okay? And since you guys deal with molecular interactions, okay, a world of molecules and microbes, okay, this way of thinking may provide a biochemical context, okay, a biochemical matrix in which you can lump all living and non-living molecular material, okay, that with which microbes interact, okay, biochemically, to change, to alter, to transform, to reshape it, and to regulate those architectures. Okay. So that's really what I'm going to say. Okay. That is, and I'll say at the end that what we might need is that omics have brought us a lot of knowledge of list of organisms as well as within cell dynamics. Okay. However, in order to deal with such a complex system as the ocean, okay, I will suggest that we will need constraints such as imaging, okay, at very fine scale, okay, going down to molecular imaging. And one revolution that has not been sufficiently integrated, okay, into our current study of ecosystems is combining omics, okay, with looking to see who is there, what it is doing, and what it is doing, hopefully, in the future at the molecular level. Can we see biogeochemistry? Can we see ecology happening? Can we see microscale interactions in action? Okay. And I would say that because of the progress, recent progress in imaging technologies, we can begin to do that. Okay. So the constraints that we can discover by simply looking, okay, added to the powerful omic techniques and approaches of systems biology may actually provide us the context in which we can begin to understand how microbes as such pervasive and powerful biological forces structure the ocean's ecosystems, and to be able to predict, because that's the name of the game, to be able to predict the shape of the ocean to come. Okay. So basically now I will tell you what time will permit to support what I have said. Yeah. There are some numbers I have told you, but to focus on bacteria interaction with organic matter, a major point that I want to make is that primary productivity is largely a particulate phase process. Phytoplankton that's produced, that you saw on that big picture, that's really particles. These are organic particles. However, we just said bacteria are obligate osmotrophs. They can only use dissolved organic matter. So we know that somehow a large fraction of primary production ends up into bacteria, but highly variable fraction. Okay? We don't really know how particulate becomes dissolved. That's why actually also I showed you the picture of those bacteria hovering around those detritus and phytoplankton cell to show that actually they use their enzymes on the surface in order to make soup out of these particles. So these interactions we generally know, but we don't precisely know. But understanding the variation in this and interaction, understanding the interaction of bacteria with the phytoplankton and particles as they tend to sink down, that's really the central theme. That's what we need to figure out. Variation in nature and strength of this coupling. Okay? Because it affects how much is going to fall down, how much is going to go to the fish, and how much is going to be respired back to this as CO2. And the challenge really is understanding how microbiology really plays out, because you look at the microbe as a species or a taxon. So really what we don't know, and we don't know it in any system, not just ocean system, is hard as microbial ecology plays out in the natural environment. Okay? And what are the consequences for the ocean ecosystem? Okay? So I give you 3 examples of why that's so important to know. We would like to predict what happens if you enrich the ocean with iron, for instance, to increase the productivity so that we'll get rid of CO2. However, we cannot predict what kind of ocean we'll end up with. Okay? A very interesting problem that some of you may not know is that most of the dissolved organic matter in the ocean is actually refractory. Refractory DOC. Something like 85% of the dissolved molecules in with which bacteria interact are actually not usable by bacteria, not used by bacteria. Their carbon age in the deep sea is on the order of 6,000 years. Okay? We don't really know why they cannot interact with these molecules. Okay? And not knowing this means that we cannot predict if a bacterium will emerge that will begin to respire them, okay? In which case the CO2 will substantially increase because the pool size of this carbon is on the order, roughly it's about 85% of the pool size of all the CO2 in the atmosphere. Okay? We don't know if we might actually be doing the reverse, that is the ocean is storing more carbon into dissolved phase. And we don't even know. We may be sequestering more carbon than we think. But understanding these microbial interactions, okay, at the molecular scale is what we need in order to understand that. So in order to understand this coupling, what are the challenges? Okay, what are the challenges for system biology, or what are the challenges for oceanographers? Okay. Primary productivity that I showed you in the beginning doesn't just fix carbon and make phytoplankton cell. It creates a huge amount of molecular diversity. And this is simply a cartoon, but actually in the real ocean, given the pool that I showed you, they're on the order of 10 to 12 estimated diverse molecules. So this interaction problem and is a very biochemically a complex problem, and the way the tight coupling between bacteria and organic matter is probably achieved is because of this great diversity of genetic diversity and biochemical diversity that comes out of it. Okay? So the interactions, because of this huge surface area and great diversity, that we can have this interaction. Okay? An important thing about the ocean that is only now coming out, and Monica Oriana has been active in this, and others in University of Washington as well, and that's the discovery about 10, 12 years ago that the ocean seawater is actually more like a thin biofilm. So it's not just seawater with bacteria swimming in it, but it's actually structured, okay, with gels and polymer crisscrossing to make a thin biofilm. You can see bacteria here. So what it does is that it makes it indefensible to assume homogeneity of the distribution of molecules in the environment. So if you make models, okay, you cannot assume homogeneity for measuring kinetics, etc. Okay. What it also does is that it creates these enormous surfaces for maintenance of diversity of microbes. Okay. So basically what I want to do is to say that we can think about the ocean with bacteria being here and their interaction with the ocean system, that interaction to be with a molecular continuum where I've thrown in people to small molecules, everything together. Okay. I'll give you quickly two examples of why that approach of studying interactions of bacteria at the molecular scale is useful for understanding at the biochemical level and hopefully modeling, okay, small scale interactions, why it's important for the ocean basin scale carbon cycle. One example is work by my ex-student K. Baidu, we found that diatoms, which as you know, silica shells are covered with a proteoglycan, he found that silica dissolution is actually not just chemistry dissolving silica, but actually bacteria exert their proteases on the surface of the phytoplankton cells, denude it, and then it dissolves rapidly. So what it is that you can then have the opportunity to model silicon cycle regulation in terms of bacteria interaction with this molecular continuum. Okay. A second example is marine snow, which has a very high concentration of bacteria, which as the particles sink, they are increasing, they're creating these plumes of dissolved organic matter with which bacteria can also interact. Okay. And the final thing that I want to point out is about this need to emphasize seeing at the small scale. Okay? And this is simply to dramatize that point that seeing really helps you reduce the complexity a little bit in terms of asking the questions. And seeing at this nanoscale, this nanoscale imaging revolution that we all know about, so that is actually making it possible actually to see the seawater at the very small scale. Okay. Using atomic force microscopy, my student Francesca Malfatti, for instance, discovered that what we had been counting as Synechococcus cells actually had a substantial fraction of cells had heterotrophic bacteria associated with them. So you have a primary producer and a decomposer tightly coupled together. And that changes thinking about carbon fixation and carbon decomposition. Okay? And another example of this tight coupling between cyanobacteria and heterotrophic bacteria. Okay? And also, it turns out that bacteria can be attached with networks and previously that nanowires. Okay? And the final point I wanted to make, which I don't think I have the time to make, was that understanding molecular interactions at a small scale has implications for human health issues, such as, in this example, Vibrio cholera thrives in hotspots of organic matter. So blooms come, Vibrio cholera can actually thrive depending on the presence of dead algae, and it doesn't attach necessarily with it, but it hovers around and can grow very fast. So when in this red tide where we got these samples of dinoflagellates and added Vibrio cholera, toxigenic Vibrio cholera, you could see these associations that can lead to proliferation. And also what it says is that knowing the ocean ecosystem at small scale may be important for understanding and modeling the ecosystem for the emergence or proliferation of human pathogens. So understanding the patchiness in the ocean is important for understanding the... so I will go to actually conclusions part because short of time. So what I've said is that we need to, there are pressing reasons why we need to understand how the carbon cycle in the ocean works. Okay. We know that microbes are very important in this process. Okay. They are abundant, they're highly diverse. We have used approaches of genetics, genomics, all omics approaches, and we know now a lot about what organisms are there. these approaches are not sufficient for understanding. In fact, we still don't know how microbes structure the marine ecosystem. Okay? However, in order for us to understand and predict the ocean's carbon cycle in the future, okay, we need other approaches, and I'm suggesting that if we converge bacteria omics with nanoscale imaging and what I've called diffusome analysis, which is that these interactions are not only with these connections that I showed you between microbes and microbes and other organisms or organic matter, but also as diffusion occurs in the example of the cholera that I showed you. So structuring the system in the space, then we can discover a mechanistic basis of this biogeochemistry. Okay? And we can use ocean models starting with simple models such as a drop of seawater. Okay? If the ocean were simply a drop of seawater, okay, and we were told that our life and our welfare depended on understanding the microbial ecology and the biology and the chemistry that goes on in that drop of seawater, we would all be sitting there with our nanopipetters, okay, taking samples to see how the system works. And we would probably figure it out. Okay? And if just had a drop of seawater, and it's a question of scale, okay, a drop of seawater, we would all say, all right, no problem. Okay? Give me a picoliter here and a femtoliter there, and I can figure things out. I'll figure out all the list of everything that's there, how it's interacting with the system, okay, and what are the consequences for gas exchanges and what goes in and what comes out, okay. So it's the enormity of the ocean system and the complexity of the ocean system, okay, that is so daunting, okay. But the question is a drop of seawater much less complex? It's already very complex. Okay? But I think that's where we can begin to have these models, and a drop here and a drop there all adds up, meaning we can have different representation of different types, and we can do perturbation experiments to do that, and then we can complicate it by adding other parts of the ecosystem. And we can models and predict future ocean carbon cycle, and hopefully also anticipate what other issues of ocean health and human health may be coming up that we need to worry about. So basically the message is that genomics and system biology approaches have been very powerful and useful for understanding the ocean's ecosystem, but we need more because this is not far enough. And what we need is one thing that we need that is available now, okay, that we need to begin to look at the ocean. Thank you very much.